Hello everyone, hope you're all doing fantastic. In this video I'm going to be continuing my series where I attempt IB Biology HL paper 1 questions on camera with you guys so that you guys can see my thought process and attempt these questions yourselves. Based on the support and feedback I received from my previous videos, I've decided to further continue this series as it supposedly helps you guys in your preparation for your upcoming exams. Without any further wait, let's get straight into these questions. Which label structure on the electron micrograph below identifies this as a plant cell and not an animal cell? Plant cells versus animal cells, the distinguishing structure you always think about are chloroplasts. And if we're going to look at a chloroplast over here, or, well, it could be a chloroplast, but chloroplasts seem to be these big structures here, which there are no arrows pointing to, so this might not be the answer. Let's think about something else that a plant cell has that animal cells don't have. Well, one thing I can think of is a large central vacuole, which seems to be this one that D is pointing at, because we can see this structure here, this super large structure, it's not as large as this central vacuole thingy. This is the nucleus, so D seems to be the correct answer. What is the sequence of stages during the cell cycle? In the cell cycle, we have interphase followed by mitosis and cytokinesis. So it goes G1, S phase, and G2. That seems to be correct. This one is incorrect, this order is incorrect, and this order is definitely incorrect. So A is the correct answer. The levels of iron in a liver tissue from 12 rats fed on beef and 11 rats fed on plant oils were compared using t-test to see if there was significant difference at the 5% level. Part of the table showing the critical values for the t-test is shown below. We have the degrees of freedom listed here. We have the p-values here. What is the critical value above which the two samples can be considered significantly different? I believe the degrees of freedom is to do with the number of samples. So it would be 12 plus 11 minus 1. So that's 23 minus 1, which is 22. So let's look at the 22 column. And if we're looking at 5% level, we're going to be looking at here. So 2.074 seems to be the correct answer that I'm going to pick. When observing the behavior of a vesicle in a cell, what identifies it as a vesicle only involved in exocytosis? Adhesion between two lipid bilayers, fusion of two membranes, secretion of materials, invagination of the plasma membrane. If it's exocytosis, it would not be invagination. So actually, why am I circling it? I should be crossing it out. Only exocytosis. Well, I think let's see if any of these options can be associated with endocytosis. Adhesion between two lipid bilayers, that happens in endocytosis. Fusion of two membranes, again endocytosis. But secretion is never associated with endo, it's always exo, so C seems to be the correct answer. Which diagram best illustrates the interactions between water molecules? We have hydrogen bonding, and this is not it, this is not it, this is not it. It's always between oxygen and hydrogen in adjacent molecules. The base ratios in the DNA and RNA for an onion are given below. A, adenine, guanine, cytosine, and thymine. We see they're basically equal. There's not equal for some reason, but they're basically equal. Yep, and we have RNA. DNA, what is the reason for the difference between these figures? Well, the difference is that when RNA is synthesized, well, RNA should not contain the same things as DNA. That's basically what this question is asking. And why these percentages vary across. Why there's more G in the RNA, more C in the RNA, but less of T and A. If you think about it, when we make RNA from DNA, for each A, we're going to have a U, right? And for each G, we're going to have an a C, and for this one, we're going to have a G, and this one's going to have an A. So we can already eliminate option C because A and T are complementary, and in RNA, A and U are complementary. DNA is made entirely of a double helix, but RNA is not. Oh well, we'll leave that option for now. DNA is only found in the nucleus, RNA throughout the cell. No, that doesn't really affect why these numbers differ. RNA comes in 3 forms, DNA only comes in 1. Again, that doesn't really show why these figures would differ, but if we look at this option again, DNA is made entirely of double helix, while RNA is not. So if RNA is single-stranded, that means that the number or the ratio, this one to this one, or this one to here, G to C, A to U, does not have to be equal, because you can have a chain of solid A's, 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 and then G, 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 G. Does not mean we need a C, C, U, 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 because RNA is single-stranded. So that sort of provides explanation for why these numbers differ. So B seems to be the correct answer. What is lactase used for? Lactase is used to hydrolyze lactose into glucose and galactose, I believe, so not fructose. It improves digestion of milk by some people. That is true. Lactose is used to form lactose-free milk. Lactase is used to form lactose-free milk, not sugar-free milk. Digestion, that's true. Acidity, no, acidity is not associated with lactase. What will be produced during aerobic cell respiration? We know water is produced, ATP is produced, ethanol is produced from anaerobic. So 1 and 2 are the correct answers. Which graph best represents the effect of temperature on the rate of photosynthesis? We gotta consider that temperature affects enzymes, and enzymes denature when temperature is too high. So since enzymes are used in photosynthesis, we can assume that the graph is basically the same as that of temperature versus enzymatic activity. EA for enzymatic activity, T for temperature, sort of like this. So it's like a reverse parabola, and C is the only option that matches that description. What does the nucleus of a human lymphocyte cell contain? Lymphocytes are blood cells, right? And they're more to do with tertiary, third line of defense. Human lymphocytes don't have genes for antigen, they produce antibodies. Range of antibodies, no. Each lymphocyte contains one antibody gene. Whole genetic information, maybe. Only genes that control growth and development, no. I believe it is D, because every single cell in the body, or every single nucleus of the somatic cells, have the entirety of the genome, but it's just that these genes sort of have not all these genes are activated. Some are repressed and some are enhanced, some in some terms of differentiation, but the underlying genetic information is present, so the whole genetic information should be present. So D is the correct answer. Chorionic villus sampling is sampling from, I believe they are cells from the fetal digestive system. I think chorionic villi is to do with the placenta. They are from the placenta, right? So I know that amniocentesis is this definition, so that's not true. And umbilical cord, no. Sampling of cells from fetal digestive system, I don't think so. I think it is the placenta that's of concern here. Because the chorionic villi are part of the placenta, they help increase the rate of absorption by having villi. How is red-green color blindness inherited? Girls inherit the condition from their fathers only. That's not true. This is a sex-linked. Boys can inherit from the condition from unaffected parents. Yes, indeed, they can, because they can have an unaffected mother and an unaffected father, but the mother being a carrier can provide, can give the son or the boy red-green color blindness. To produce artificial erythrocytes used in blood transfusions, tobacco plants have been genetically modified to produce human hemoglobin. The first three triplets of human hemoglobin are ATG, GTG, and CAT. What would be the first three triplets if the hemoglobin gene inserted into genome of modified tobacco plants? Well, if this is the gene and we're inserting it to the genome of tobacco plants, wouldn't it be the same gene? I'm not sure, but I think it's basically the same gene that we're putting in. If this is a gene that we need for human hemoglobin and we're putting it to tobacco plants, we're going to put this into that, so it's fine. What is the energy transfer level from the kangaroo rat to the weasel shown in the food web below? Bobcat, roadrunner, gopher, snake. Okay. Kangaroo rat is this one right here. Weasel is here. 3 times greater than the energy transfer. Roadrunner to the bobcat. Well, I believe that only 10% of energy is passed on, so the 3 times does not really make sense here. meadow mouse to the opossum. Okay, so I read through the options. Quarter of the energy, that doesn't really make sense. Half the energy, no. Again, the 10% relationships don't really allow for these calculations. And since this is a paper one, I doubt there'll be any 3 times, half times, quarter times without use of calculator. But option D does seem promising because we have meadow mouse, a primary consumer, just like the kangaroo rat, being consumed by a secondary consumer, just like the opossum and the weasel. So meadow mouse or opossum seems like the same. Seems like the correct answer. Which of the following gases will contribute to the greenhouse effect? Oxygen, nitrogen oxide, argon. I know that oxygen does not contribute to greenhouse effect. It's pretty good as a gas. We need it, and it's not really considered a greenhouse gas anyways. Argon is also not discussed, so you can eliminate A, C, and D, so leaving B behind as our correct answer. Why has antibiotic resistance evolved in bacteria? All bacteria reproduce very quickly. That's not really to do with resistance. Bacteria exposed to antibiotics, no, it's not that bacteria exposed to antibiotics, they don't develop resistance, they have resistance. It is that bacteria showing these resistant traits will survive after antibiotics are used. Which taxa do Zorinthia rumina and Zorinthia polysina share? They share the same class but not the same family. Well, kinder, phylum, class, order, family, genus, and species. This is the genus, this is the species. They have the same genus but they're not the same species. Um, There's no option like that, but we know that they are the same genus, so this is not correct. They are not the same species, so it could be these two. They are the same family because if they, so it goes kingdom, phylum, sorry I spelled that wrong, phylum, class, order, family, genus, species. I just remember that from the top of my head. If we have genus being the same, that means all these are also the same. So if we have the same genus, they are in the same family, so A is incorrect. If they are in the same genus, like we identified here, because this is the genus, this is the species, then they are in the same class as well, because that's above. So B is incorrect. They do share the same family, because again, they share the same genus. They're not the same species, because this one is rumina, this one's polysina, so D is correct. What features distinguish Platyhelminthes from Annelida? Platyhelminthes, I believe, they both have bilateral symmetry, so we can get rid of these options. I believe Platyhelminthes do not have a segmented body. Annelida are like earthworms, and I know for a fact that they have those ridges that sort of segment them, so B might be correct. What are the names of the organs labeled 1 and 2 in the diagram below? One is definitely a tiny piece from the liver, tiny looking piece, so this is the this should be the gallbladder, right? And this big thing is the pancreas. So 1 and 2 are the gallbladder and the pancreas. So C seems to be the correct one. Where in the cell are antigens found? Antigens are found on the plasma membrane. Which feature maintains a high concentration gradient of gases in the ventilation system? I believe it is the concentration gradient in the ventilation system. The concentration gradient should not be based on thin-walled alveoli or thin-walled capillaries. That would actually help with movement of gases, so this doesn't seem right. The moist lining of the alveoli, again, I don't think the moist lining would affect it, but I think the blood flowing into the capillaries, that sort of exchanges gases, and that might be what maintains the high concentration of gas gradient. What causes the reformation of nerve impulse on the postsynaptic membrane is Ca2+. Neurotransmitter being removed from, I believe it is to do with Ca2+. I'm just considering whether it could be, well, it's definitely not cake, this option, because there's no leaking into the postsynaptic membrane. Neurotransmitter binding with receptor sites, I believe that does happen, that binds with neurotransmitter receptors on the postsynaptic membrane, but Ca2+, maybe it is Ca2+ that binds with the receptor site. I'm not really sure. Oops. I'm not really sure. Neurotransmitter binding with receptor sites. Neurotransmitter being removed from synapse. That's not it. Let's consider this. So receptor sites. I feel like Acetylcholine receptors are to do with neurotransmitters, whereas this receptor is a general term for receptors. And when the neurotransmitter does bind, you get CO2 plus flowing in that binds to further receptors that open sodium channels. So I think A might be the correct answer for this question, because the sodium plus moving is the nerve impulse itself. And right before that, the previous, like the directly previous event is the CO2 plus binding with receptor side. What is the difference between the origin of type 1 and type 2 diabetes? In terms of origin, type 2 is generally late onset, so this is the one where you eat too much, target cells fail to respond to insulin. This option A seems to be correct. This is not correct. This is not correct. This is swapped around, again swapped around, and again swapped around. So A is correct. The hormones progesterone and luteinizing hormone LH were measured in a woman's blood over 40 days. When did her menstrual bleed start? Well, if we're talking about menstrual bleeding, that's talking about the period and when menstruation occurs. When menstruation occurs, we're shedding the inside of the uterus. And when females do this, it's generally because the vascular endometrium cannot be maintained because there's not enough progesterone. So we should look at places where progesterone levels are really low. And I also believe that when progesterone levels are really low, luteinizing hormone is really high. And luteinizing hormone is important for the formation of corpus luteum. And this is generally where this should be near where menstruation occurs. So we look at where the two of these mentioned characteristics occur. B is not correct because progesterone is high. I don't think. Again, A is also probably not correct because progesterone levels are relatively higher compared to point C and point D. I would be tempted to go with point D, but it's just that we have this long period of low LH, and this is right before this peak, so I feel like this is to do with some other part of the menstrual cycle, and the period should be about here, I believe. Menstrual bleed should be here, so C seems to be the correct answer. And this topic is quite complicated about the menstrual cycle and how the hormones work. If you guys do want a video on that, please write a comment down below, and that'll remind me to get working on that. I make videos upon request in this channel, so it's generally like what I do for you guys is based on what you want. So please just let me know what you would like to see, and I'll be happy to respond and make a video on that. How does transcription of RNA proceed? 3' to 5'. Exons and introns are used, and it's always 5' to 3'. Which of the following correctly shows a peptide bond between two amino acids? These are amino acids. These seem fine. No, wait, this is not it. This is not fine. It's between nitrogen and carbon, so this one's okay. This one's not okay. C is the correct answer. What is a polysome? Polysome is a ribosome that is synthesizing proteins from several mRNA molecules at the same time. I believe it's not one ribosome, it's many ribosomes, as the polysome, polyribosome name suggests. So several ribosomes using mRNA molecule to synthesize protein at the same time. That seems to be the correct answer. What does oxidation involve? This is basic chemistry, loss of electrons. What is required for ATP synthesis in mitochondria? Active pumping of protons into the matrix. It's not into the matrix, it's into the intermembrane space. What happens in the light-independent reactions of photosynthesis? This is with the Calvin cycle. ATP is not synthesized. Water molecules are not produced. Reduction of CO2. I believe reduction of NADP would mean NADPH is formed, but I'm pretty sure NADP is produced in the Calvin cycle of light independent reactions, or stage 2 of the photosynthetic stuff, both from in photosynthesis and NADPH reduction, NADP reduction to NADPH. I'm pretty sure that happens in the light dependent reaction. So this is not it. D must be the correct answer. Which two tissues of a leaf are photosynthetic? The epidermis is not. The xylems are definitely not. Palisade and spongy mesophyll cells are definitely true. It's just palisade mesophyll cells is, I believe, one of these is more dense than the other. I don't think you need to know specifically in the syllabus. You just need to know. I believe that the palisade mesophyll is the super chloroplast dense cell. So these are the ones that are mainly photosynthetic. So the spongy mesophyll is a lower layer. They don't have that many. They don't have that many uh, chloroplasts, so not as much photosynthesis. But nonetheless, they will do photosynthesis. So B is correct. How are fluids transported in the xylem and phloem? Xylem away from root only towards the root. I think xylem is always away from root because xylem always connects root to the leaves. So this is xylem away from root only, makes sense. Phloem can be towards root or away from root, based on where stuff is synthesized. So based on season as well, I think they can change. So I'm thinking D, maybe. Yeah, because I remember a specific question asking, maybe my teacher asked me this, or I asked my teacher, or someone else in class did. It was about whether certain parts of a plant can act as a source and a sink, based on different periods, and I was told that they can, in terms of season. Sometimes the source and the sink can be reversed based on seasonal aspects of a plant. So I think away from and towards the root can work. How does the phytochrome control flowering in plants? PFR turns to PR in light. No, that's not true. It's always PR to PFR in light. So PR to PFR in light. So this one seems to be correct. But actually, PFR to PR in the dark is also correct. So it's too early to rule this out. Long day plants to flower. Well, let's think about this. If PFR converts to PR, right, in the dark, there's going to be a lot of PR in the dark. And long day plants don't have a lot of dark. So PR being a lot of dark would not favor a long day plant. So C seems to be incorrect and B is the correct answer. Let's see. PR turns into PFR in light, and long day plants, well, if it's a long day, there's going to be a lot of light, and this reconversion back to PR is not going to be as high. So we're going to have this super high concentration of PFR, and that seems to be what causes long day plants to flower. So B seems to be correct. Which process results in recombination? Meiosis. Yes, meiosis does. Crossing over, yes, it does. So B and D are eliminated. Independent assortment. Ooh, independent assortment. I believe independent assortment means that the genes are not linked, and I believe for recombination you need genes to be linked because we need chiasmata forming between homologous chromosomes in these bivalents, right? So if we want that to occur, we're going to need non-independent assortment. So C seems to be the correct answer. What is the correct sequence of factors involved in blood clotting? Platelets, clotting factors. This is skipping thrombin to prothrombin, but I guess that's fine because all these seem to be skipping that step. Platelets do release clotting factors, not the other way around, and we need have fibrin to fibrinogen, so A seems to be correct. Oh, wait a second, no, fibrin, wait a minute. I'm trying to remember. I believe it's the shorter form. Fibrin. Oh yes, fibrin is used as an example of a protein as a clotting factor. So it has to be fibrin. That's the final product that does the blood clotting and forms a mesh. It is a fibrin mesh. Yeah, yeah. It's a fibrin mesh that formed, not a fibrinogen mesh. So C seems to be the correct answer, not A. The problem there was it's a bit confusing to remember fibrin and fibrinogen, and which one is converted to which. How do vaccines give immunity to infectious diseases? It's not, they don't kill pathogenic microbes. They produce an immune response, that's true. They don't contain antibodies, not all of them. They inhibit antigen antibody reaction, that's not true. That would actually worsen your susceptibility to infectious disease. What are the names of the two structures labeled 1 and 2? 1 is definitely the tricep. Let's be real, we all hit the gym and we know the triceps. Number 2, this is the radius and this is the ulna. Radius is always on top, so ulna seems to be the correct one for this. So D is the correct answer. What does the reabsorption of glucose in the proximal convoluted tubules of kidney require? I believe it's active transport. I mean, if it's reabsorption, it shouldn't be moving to water. Active transport, K plus, facilitated diffusion. Maybe it's this seems to be an old syllabus question. I don't think this is in the new syllabus, but I'm gonna try and attempt it anyway. Actually, I've been thinking for quite a while now, and I don't think I can figure this out. I generally don't think this is part of the syllabus, so I'm just gonna eliminate this question. I know it seems like a cheesy way to do this, but if you guys do think this is in the new IB syllabus, IB biology syllabus, please let me know in the comments down below, and I'll make a new video where I do attempt this question. Or if you have an explanation and the answer to this question, please just write it in the comments below and help everyone out. What is the role of FSH in spermatogenesis, secretion of testosterone? FSH in spermatogenesis. This is follicle stimulating hormone, I believe, and I believe you need FSH for testosterone to start working, right? So secretion of testosterone is done by Leydig cells, I believe. So it's not. This can't be correct. And it inhibits. No, it doesn't inhibit anything. It stimulates, and I don't think it's to do with luteinizing hormone so much in the sperm. So I believe C is the correct answer, and it makes sense because FSH, what it does is it activates testosterone, which in fact affects Sertoli cells, and Sertoli cells are those big cells that have um, a site where the sperm basically mature, and maturizing sperm gain all these characteristics that enable better ability to fertilize the egg, such as such as mitochondria, such as a tail, etc. What is the difference between spermatogenesis and oogenesis? Spinal cells are all similar in size. Spinal cells are not all the same. That seems to be correct. Cells produced are undifferentiated. Cells produced are differentiated. That's not incorrect. I think both are undifferentiated cells. Spermatogenesis begins in a boy at birth. That's not true. Spermatogenesis or sperm production happens after puberty or during puberty for spermatogenesis, whereas oogenesis is prenatal or before a girl is born. One germline epithelium cells produce formed sperm cells in the testes. That's correct. No, wait, yeah, it's correct. One germline epithelium cell produces and one oocyte in the ovary. I mean, this is not really looking at a difference, but I think it's more to do with the fact that we have the ovary as the site, and here we have the testes, but it's more the seminiferous tubules that are recognized as the main site of spermatogenesis. So I think this question is trying to get into that. And if I had to pick between A and D, I would probably pick A for this question because similar size and not all similar size, because oogenesis forms polar bodies, right? You're gonna have three polar bodies, big egg cell. And spermatogenesis, you just have all sperm that are like really small, same size, similar size. So A seems to be the correct answer. Now it's time for me to look over the mark scheme and see how many questions we got right. Okay, so upon looking at the mark scheme, it seems that we managed to get 36 out of 40 questions, and that includes a mark down for the question we skipped. So this seems to be a pretty good score because this is like 90%. Now let's go on and check out the questions that we got right. Okay, so for this question, turns out the degrees of freedom was actually 21. So the P 0.05 was correct, but I was meant to look at this option, so B was the correct answer. It's 21 because you have to subtract one from each class, I believe, but I'm actually not sure why it's 21. The answer option said that, and there was no real explanation. If you guys would like me to make a video on the chi-squared beyond the one I made already, actually, I think in the video I made, I just forgot right now. I'm really sorry about that, but on the video I made about chi-squared, which you can find on my channel or on the top right now, you can click on that and learn about chi-squared. I'm pretty sure that provides an explanation for this question and why the answer is B, 2.08. What causes the formation of a nerve impulse on the postsynaptic membrane? It was a neurotransmitter, I believe, binding with receptor sites. The formation of the nerve impulse and the start, I think, is to do with Ca2 plus, but I don't think they bind to receptors. I think they just flooding of Ca2 plus actually causes threshold potential to be reached, which then triggers the opening of sodium channels and the continuation of nerve impulse. So A would have been incorrect and C is the correct answer. Which process results in recombination? So apparently A was the correct option, and independent assortment is needed for recombination. And I think it's more that it's not really independent assortment being needed. I think independent assortment doesn't really affect recombination at all, but it's the fact that answer option C said that mutation results in recombination, which is not true. So it's definitely not true. Recombination is basically the production of, I think you can refer to the prophase 1 recombination and also the formation of recombinants, which are basically traits that are not present in parent populations, and independent assortment of genes can form that. But it's mainly the idea that mutation, it's not a yes, mutation is a no. So mutation does not result in recombination of genes. So I should have eliminated these options earlier. I went from left to right instead of considering which options are the obvious indicators of distractors. So that's a good strategy to utilize. Don't always go from left to right in a table. Check out which options might be correct, which might not be, and that way you can have the best chance of getting the question right. So hopefully you guys found this video helpful in your preparation for your IB biology exams, and if you guys did so, please like and subscribe. It will really help out the channel. It's so appreciated by me, and I will continue to deliver good content to you guys. If you have any feedback or future video suggestions that you guys want me to make videos on, like say topic 11.4, you guys can just leave it in the comments down below, and I'll make sure to get to that. Other than that, hopefully you guys enjoyed this video. See you later. Peace out.